Imagine this. What if bacteria weren't mere mindless invaders, but actually had a memory of their past? One they could pass down to their descendants. That's exactly what an eye-opening study from the Hebrew University of Jerusalem has uncovered. Using a pioneering approach known as Microcolony-seq, scientists have revealed that single bacterial cells can carry hidden memories of their environments, which shape the behavior of future generations. Until, surprisingly, these memories are wiped clean. This discovery could transform how we understand persistent infections and why standard treatments sometimes fall short. But here's where it gets controversial. Could our entire approach to antibiotics and vaccines be missing the mark by not accounting for this unseen microbial history? Let's break down this breakthrough. For years, scientists observed that even genetically identical bacteria behave in radically different ways. Some grow rapidly, others languish. Some survive antibiotics, while others perish. Yet it remained a mystery. Were these differences random flukes? Or did they hint at something deeper, perhaps even heritable? Enter Microcolony-seq, a novel method developed by Dr. Faigenbaum-Romm and her team under the guidance of Professor Nathalie Q. Balaban. Along with Professors Elin Rosenshine and Masket Bar-Mer, this technique isolates tiny bacterial microcolonies, the earliest sprouts from single cells, and meticulously analyzes their RNA, DNA, and physical traits. By doing so, it strips away the noise often encountered in even the latest single-cell genomics, providing a clearer view into whether variations are due to permanent genetic mutations or inheritable but reversible states, known as epigenetic changes. Here's the part most people miss. These memory traits aren't just academic curiosities. They have real-world consequences. The team found that dangerous bacteria like Escherichia coli and Staphylococcus aureus, even within the same infection, split into distinct subpopulations. Some groups turn on genes that help them attack host cells, while others kick off survival programs or enhance movement to escape threats. Even more intriguingly, these inherited survival strategies can persist for up to 20 bacterial generations. But, and here's the twist, once the bacteria run out of nutrients and enter what's called the stationary phase, their collective memory gets erased. The colony resets, losing the historical baggage of past environments. This phenomenon has profound implications for healthcare. Take urinary tract or bloodstream infections as an example. Microcolony-seq found that within a single infection, multiple bacterial subgroups exist. Some more resistant to antibiotics, others more virulent. Standard lab tests often examine just one of these groups, potentially missing the silent survivors that cause treatment failure. That tricky detail alone could reshape clinical practice. Are we treating infections with incomplete information, dooming some therapies to fail from the outset? Professor Balaban puts it plainly. An infection isn't a single uniform army. It's more like a team of specialists, each with unique roles and strengths. If we want to defeat them, we must understand and target every player. This idea may ruffle feathers, especially since many recent drugs and vaccines against S. aureus have failed in trials, possibly because they focused only on dominant subpopulations, unintentionally leaving others free to thrive. Peering further ahead, Microcolony-seq opens doors for exploring many forms of microbial life, from uncovering how pathogens hedge their bets between virulence and dormancy, to mapping the complex ecosystems in our gut, and even fine-tuning industrial fermentation processes. Dr. Faigenbaum-Romm sums it up best. For too long, we've treated bacterial populations as uniform, when in truth, every single cell is shaped by an invisible history that can influence the fate of millions. So here's a controversial question. Should we be rethinking how we test, treat, and even talk about bacterial infections? Are current clinical methods dangerously outdated in the age of single-cell science? Let us know what you think. Does this groundbreaking discovery challenge your assumptions? Or are there hidden risks in embracing it too quickly? Join the conversation and let us know where you stand.